Hello students, welcome to BioSmart Academy. In today's lecture, we are going to discuss about the final two types of hypersensitivity, that is type 3 hypersensitivity and type 4 hypersensitivity. So before starting these two lectures, I just want to remind you that please subscribe BioSmart Academy because this YouTube channel provide you all important lectures on CSI net syllabus, strictly on the base of CSI net syllabus, and within the month of December you will prepare yourself for this all examination, that is NET, GATE, DBT and ICMR. So please subscribe this channel and please share this channel as well to all those students. Please share this channel to all those students who are preparing for all this examination, because they need these lectures very much, because this YouTube channel provides all the lecture according to the CSI net syllabus. So please share and subscribe BioSmart Academy as much as possible so that you can prepare yourself for the December 2020 CSI net. You will definitely get a good result if you follow BioSmart Academy. So let's start today's topic, that is type 3 hypersensitivity. First we look at this and then we look at the most important, that is type 4 hypersensitivity. Okay, so let's look type 3 hypersensitivity. The most important thing that which immunoglobulin basically takes part majorly in type 3 hypersensitivity, that is IgG. Okay, in type 1 it was IgE, I think you remembered all. In type 2 it was IgM and IgG. In type 3 it was IgG. Okay, so question may be asked that type 3 hypersensitivity is mediated by, so the answer will be IgG. Clear? And it is mediated by immune complex, essentially of IgG antibodies, as I already mentioned, with soluble antigens. Okay, and all those immune complex are basically lodged at various sites, and on all that sites they basically activate what? They activate the complement system. Okay, they basically activate the complement system and cause this cell lysis, it means they basically circulate, and after circulating they lodge at particular site where there are basically antigens are present, and there they activate the complement system and the lysis of cell is takes place. Clear? Next, the complex are basically phagocytosed. Okay, the complex are basically phagocytosed by the fixed monocyte and macrophage. Okay, and those complexes are basically phagocytosed by the macrophages and monocytes. Clear? So basically type 3 hypersensitivity was first mediated by IgG. Okay, and then the immune complex are basically circulate circulate at various and lodged at various sites, circulates and lodged at various sites where they activate the complement system, and after activation of complement system the lysis of cell takes place, Clear? And these complexes are basically phagocytosed by the fixed, which are a fixed monocyte and macrophage. Clear? Next, in type 3 hypersensitivity, as I already mentioned you that it was IgG immunoglobulin mediated. Clear? Next, so when there are excess soluble antigens are present, okay, excess soluble antigen are present, then the antigen antibody complex is not efficiently removed all that soluble antigens. Okay, they are incapable of removed all that soluble antigens, so their accumulation lead to hypersensitivity reaction from the complement system, and complement system basically triggered the inflammatory process. Okay, then the complementary system triggered the inflammatory process. Clear? And mainly the antibody involved here is IgG. Please remember this point. So this is all about type 3 hypersensitivity. Okay, this type 3 hypersensitivity is not so much important from examination point of view. I will discuss that from which type of hypersensitivity most of the question has been asked previously, and future the question will be asked. Okay, So please watch this video till end. Now we are going to discuss the most important, that is type 4 hypersensitivity. Okay, first of all, I marked all the important points. Please remember. Okay, first of all, type 4 hypersensitivity is known as DTH, that is delayed type hypersensitivity. Okay, type 4 hypersensitivity is known as DTH. Okay, DTH, that is delayed type hypersensitivity. Please remember, question may be asked that which type of hypersensitivity are known as delayed type hypersensitivity or DTH. Okay, so delayed type hypersensitivity is basically type 4 hypersensitivity. Clear? Next, type 4 hypersensitivity is the only class of hypersensitivity that is not triggered by any antibody, or you can say any immunoglobulin. Okay, not triggered by any immunoglobulin. This is totally mediated by cell mediated immunity. Okay, this is totally mediated by cell mediated immunity, means this was mediated by CMI. Okay, this was mediated by CMI, that is cell mediated immunity. So this is the only class of hypersensitivity which was mediated by cell mediated immunity. It is not dependent on immunoglobulin. Clear? Question may be asked from this point as well. And the third point is that this is mediated by cell mediated immunity, mainly the involvement of TS cell and especially of TH1. Okay, especially of TH1 cell. Okay, please remember, especially of TH1 cell. And I discussed about TH1 cell in details in the previous lecture. Please go and watch the cells of the immune system lecture, in which you will find out all these things. Clear? Next slide. See, as I already mentioned that antibodies or immunoglobulin do not play any role in type 4 hypersensitivity reaction. So please remember this. Type 4 hypersensitivity reaction is basically mediated by CMI. What is CMI? CMI is cell mediated immunity. Clear? Next, I already mentioned that TH1 play a very important role. And what is TH1? You will find in my previous lecture of cell mediated immunity. So you can watch there. Okay. So basically TH1, what is the function of TH1? TH1 basically release cytokines. Okay. TH1 release cytokines, and these cytokines basically accumulate. Okay. These cytokines accumulate there, and basically these cytokines lead to the activation of macrophages, which cause local damage. Okay. It means TH1 basically activate cytokines, and cytokines activate macrophage. Cytokines activate what? Macrophage. Cytokine activate macrophage, which cause local tissue damage. So this is the mechanism. Okay, so this is the mechanism. Okay, clear? Next, the best example of type 4 hypersensitivity is tuberculin test. Okay, see, question may be asked from this, which is the best example of type 4 hypersensitivity? So answer will be tuberculin test. Tuberculin test is an example of type 4 hypersensitivity. So now look at this tuberculin test. What is tuberculin test? See, this test is done by putting a small amount of tuberculin purified protein derivative. Okay, means a protein is derived from the tuberculin, and that protein is put on the layer of skin. See, suppose this is a layer of skin. Okay, then this tuberculin protein derived is put on this layer of skin. Okay, now if a person is exposed to mycobacterium tuberculosis, okay, or earlier he was exposed with this bacteria mycobacterium tuberculosis, then within 2 to 3 days, this tuberculin protein derivative is basically becomes a red bump. Basically it becomes swelling, and the color is basically red bump like this. Okay, so this is tuberculin derivative test. Okay, it was done on the skin. Okay, this is skin. Okay, this is skin, and that was the tuberculin derivative protein in tuberculin protein. Okay, and this is the red bump which was occur after 2 to 3 days. Red bump. Okay, red bump. I think now you get clear. Now let's read the PPT. See. Now, this tuberculin with purified protein put under the top layer of skin. Okay. Now, if any person ever exposed to Mycobacterium tuberculosis bacteria, then skin will react. Okay, skin will react to that antigen and develop a red bump, as I explained you in the figure, within 2 days. Okay, within 2 days, as I explained you in the figure, within 2 days that area become red bump. Okay, that area become red bump. So it is a standard method of determining whether a person is infected with tuberculosis, Mycobacterium tuberculosis, or not. So this is the experiment that was done. Okay, that whether a person was infected with Mycobacterium tuberculosis or not. I think now you get a clear idea about the tuberculin test. Question may be asked from this portion as well. Okay, so students, that's the end of the topic hypersensitivity. Okay, topic hypersensitivity. If you have any doubt on any one of the hypersensitivity topic, either type 1, type 2, type 3 and type 4, then you may ask question on the comment box. And I want to discuss with you that type 4, type 2 and type 1 is the most important one. Type 1, type 2 and type 4 hypersensitivity is the most important one, and question has been asked many times from this topic. So please read all this very carefully, and tomorrow we will start a new module, that is cell biology. Okay, cell biology, and I start cell biology because this module is very much important from CSI net point of view. Okay, 3 to 4 topics of immunology remain there, and I will cover all these 3 to 4 topics within few days. Okay, and before that I start cell biology from tomorrow. Okay, so be ready. Okay, and please share as much as possible this channel. Thank you.